All right, so today we are going to be focusing on the CMC section, which is the Chemistry, Manufacturing and Control section of regulatory submissions. Whether it is for IND or for NDA or BLA, there is always a CMC section involved, which talks about, of course, manufacturing and quality aspects, the operational aspects of producing the drug. And because our biotech program is focused more on manufacturing of drugs, we will focus on this section today, right? So just to reiterate what we already learned about the industry, if you recall, these lines are the connective tissue, so to say. It's the institutional arrangements between different entities, right? And the ovals or circles are the rules, and the rectangles or squares are the players or the participants in the whole industry. Okay, so today we are going to be focusing on this aspect, regulations from the FDA, EMA, etc., especially concerned with the CMC sections. Okay. So what we want to do today is to build on what we started talking about last week. I think in the textbook, the CMC section, which is partly covered in sections 8.1 through 8.4, it's not covered in great detail because the book is not focused on manufacturing, but our biotech program is focused on manufacturing. So we will go into a little more detail than the book does. Especially we will be focusing on larger molecules. Okay. So we will start out with the what we talked about last week, which was the ICH and the CTD in particular, which covers the CMC section, but it doesn't focus primarily on biotech product. It's a general document. So I will try to focus more on the biotech products today as it relates to the CMC section. Okay, so we will focus mostly on several areas of the regulatory interest, agency interest for biotech products in early phases of clinical study. So mostly on focus on IND requirements. Okay. So I covered this a little bit last week, a little more detail now. So the FDA has several branches, and the two that are more important to us are the CDER, Center for Drugs Evaluation and Research, and CBER, which is the Center for Biologics Evaluation and Research. In the past, CBER used to do all biotech products, but now most of the biotech products, which are biologic therapeutic products, including monoclonal antibodies, cytokines, growth factors, and toxins, they are now covered by CDER. So most of the time, we will be dealing with CDER, although CBER is involved in approving vaccines, in vitro diagnostics, and drugs composed of human or animal cells, or blood and blood products. Okay, so those are still covered by CBER. So that division is also relevant. And of course, we talked about devices and radiologic health and veterinary medicine. These are other divisions of FDA that we don't normally work with. Okay, so moving forward. If you remember, in our alphabet soup, we have three important ones, three important acronyms: IND, NDA, and BLA. IND is the application to the FDA, investigational new drug application, when we want to begin testing in human clinical trials. So that's the first submission that we send to the FDA. Then NDA or BLA, you either do an NDA or a BLA, depending on whether you have a small molecule or a biologic. So if mostly we'll be talking about BLAs because we are concerned more with biologics, right? So these are the different submissions to the FDA. How about, so just to reiterate, in our timeline, IND comes first, right after the preclinical studies. So you submit an IND to the FDA and wait for them to approve clinical trials. So FDA is involved here, and when they approve, you begin phase 1, 2, and 3 trials. And then the next big submission. Of course, you are still consulting with the FDA at each stage and even in between. But the next big submission is the BLA or the NDA. And once you get approval for that, this is usually a fairly long time, something like 12 months. After the review, if they approve, then you launch the product. Okay. So what about the European agencies? So European agencies, you might have heard of two different acronyms here. You have EMEA. So EMEA is a former name. It used to be called European Agency for Evaluation of Medicinal Products. Okay, European Agency for Evaluation of Medicinal Products. That was the former title. Now we just call it EMA, or European Medicines Agency. So that is the overarching agency in Europe which oversees the centralized process for marketing submissions. Now, if you remember from last week, the centralized process is required for all biotech products, right? Required for all biotech products. And also new drugs for some specific diseases and orphan drugs. So this is required for approval. So this is when you are applying for approval, marketing approval, right? But there are two other processes in Europe. One is called the decentralized process, in which you could apply to individual countries. And another process is called, if you remember, another process is called the, what is it called? Mutual recognition procedure, right? Mutual recognition procedure. And so mutual recognition procedure, that is when you apply to one country, but if they are part of a treaty with other countries, then you can use the same approval to apply to other countries, right? So mutual recognition procedure is the third one. So there are three possibilities, but for biotech product, you always use the centralized process. Now, on the other hand, for submission for clinical trial approval, you cannot use the centralized process. So for clinical trial approval, you have to use the decentralized process. It used to be called clinical trial applications that you send to individual countries where you are going to do the clinical trials, where you are actually going to conduct the clinical trials, right? Now, this has been largely superseded by what is something called CTX, which is clinical trial exemption. Okay, clinical trial exemption. So that's what we use now. And again, it is a decentralized process, and you have to apply to individual countries wherever you are conducting the clinical trials. Okay? And you have to submit the IMPD or Investigational Medicinal Product Dossier, which includes the clinical protocol, safety studies, and the CMC section. And again, it is in the CTD format, right? Remember the CTD format from last week? All right. So there are essentially similar procedures with some country specific country-based requirements for other countries like China, Japan, India, etc. In particular, in Japan, you have to apply to the Ministry of Health, Labor and Welfare. In Japan, it is the Ministry of Health, Labor and Welfare, right? So this is how we submit to Europe and Japan, right? So to the EMA through the centralized process, or to different countries via using the CTX. Any questions so far? Okay, so let's start talking about biologics, because as you know, biologics are very different than small molecules. So today we are going to focus on biologics. First of all, in manufacturing, there is the process is complex because we use living cells, we use living material, right? And so because living material is inherently variable, okay, so inherently variable, there is more complexity. We cannot absolutely predict what the results are going to be from batch to batch. So normally we deal with ranges. So for example, we might have a range of cell concentrations that is acceptable to us. We may have a range of product concentration that is acceptable to us. So we have to deal with this process complexity in some way. Also, because the process is complex, we cannot rely on the product testing alone, because we have to make sure that we try to make the process as consistent as possible. There is sensitivity to change and environment. For example, the product is labile. If you change the temperature a little bit, it may degrade. And the inherent variability of the process that I talked about. Okay, and then there are impurities. There is possibility of impurities, for example, contamination, and also the raw material may have viral or bacterial or fungal or transmissible spongiform encephalopathy agents, right? So there may be all kinds of impurities, and it is difficult in defining and quantitating these impurities. So again, this calls for control of process, okay, and also testing, right? So this is very different from small molecule materials, where the raw materials are usually well characterized and under control, right? So you also could have multiple species involved. Active ingredients can be of varying complexity. For example, a lot of the products, although they are proteins of specified amino acid sequence, they often have sugar molecules attached to them, right? So, and this addition of sugar molecules, which is called glycosylation, it is variable, inherently variable. You may have different amounts of sugar added onto the protein. So that is why you have heterogeneity. You have heterogeneity because of the addition of sugar molecules. So, and in addition, you may not be able to completely purify out some of the impurities. So you will have additional things in your product, right? So active ingredient is also complex. And characterization is difficult. Characterization is very difficult because the methods are, they are still evolving. Methods of characterizing protein products are still evolving, you're essentially limited in how you can characterize the product. So, for example, I'll give you one example from my past experience. We used to work with a product called Epogen, and we had characterized the product using the best available methods. So especially for the characterization of the glycosylation of this product, we used to use something called the Western blot, which is a way of characterizing proteins and different isoforms of the proteins, right? And later on, we made a submission to the FDA based on Western blot. And later on, a new method came along, which was based on HPLC. And this method could characterize glycoproteins to a much more detailed level. And what we found was, based on this method, a new process that we were evaluating had slightly different product. Had one particular sugar that was different. So you can see how, even for an approved product, you may get difficulty in characterization as new methods become available. So method limitation is a cause for concern. Right? So what I'm trying to say here is that the biologics products are much more complex than small molecule products, and therefore the CMC section for biological products has to be that much more thorough. And one of the things that I want to make sure everyone understands is that we cannot simply base our CMC section on analysis of the product alone. So analysis of the product is not sufficient. And I'll give you another example of why that is so. And I want to emphasize this because several people got one of the quiz question wrong. That the question was about whether analysis of the product is sufficient or not, and it is absolutely not sufficient. Okay, I'll give you an example. For example, we were producing a product that by every analysis that we had performed looked the same when we were doing clinical trials and when we scaled up the product to produce lots for the phase 3 conformance lots. So this was product that was going to be sold, right? So all the assays were showing the product to be equivalent. Okay, but then the way it turned out, that the product that we were producing at large scale was actually degrading over time. And so in the stability studies, we found out that the product was more susceptible to degradation than the product that we had produced for clinical trials. Isn't that amazing? The product which looks the same is actually degrading faster than the product we had used for clinical trials. Well, it turned out that when we went from small scale to large scale, small to large, what we had done was we had changed one particular aspect of the process. We were actually using a UV light to we were using a UV light to detect the product in the large scale, which we didn't have in the small scale. So this UV light made enough of a difference that every molecule that was passing through that UV light became more susceptible to degradation. Okay, so here is an example of how the process can affect the product. And even though the product looks the same, is characterized the same way, it is different. So this is why we want to make sure that the process is well controlled and we use the same process again and again and again. And I cannot emphasize this enough. The process defines the product. Okay, so it is not enough to just test the product, test quality into the product. You have to make sure that you show to the FDA that you are actually controlling the whole process at each stage. Okay, so thorough description, characterization, and testing, starting with source materials, components used throughout manufacturing. Okay, so we are not just testing the product, we are looking at everything throughout manufacturing. Greater reliance on manufacturing process control. Every step you need to specify what the limits are, right, of acceptance limits, because final product testing is insufficient to ensure quality. Okay, so careful description and evaluation of manufacturing changes made during development for potential product impact. I just gave you an example of that, right? So it's really, really important to know that the process defines the product and not just the product testing. Okay. So when FDA reviewers are looking at your submission, they're going to be asking these questions, especially as it relates to biological products. So, is the product unstable? Okay, so they have to see stability studies under different conditions. Has there been substantial amount of time between manufacturing steps? For example, you might, after fermentation, you might store the product as a frozen cell-free extract. Okay, and then you might use that product after several months for purification, depending on your production scheduling. Okay, so what happens to the product while it is being stored in between process steps? Okay, you have to make sure that you validate that the length of time that you have, you're going to be keeping the product in between, right? Was the product that was used in preclinical study different from the ones that was used in clinical studies? I think I may have mentioned this particular aspect to you. An example from Genentech, TPA, for example. They had used a different process during preclinical studies and during clinical studies. And so the FDA held up their approval for TPA because they had to show equivalence of the product, that it was similar. Okay, so these are all important questions. Are there potential safety issues as they relate to the quality aspects of the product. For example, is there a potential of BSE being included? Are there viral contaminants in there? More questions. So the type of product or the product class might affect the type of information that you need to assess safety. So there are some products that have unusual complexity. What's an example of a product of unusual complexity? Gene therapy product. Because the reason is gene therapy products they don't just go into your body and get out of the body, right? Most of the biotech products are, they impact body functions and then they get out, right? They're excreted or they are degraded or something. But gene therapy products stay with you for a long time, right? Because you're introducing genes that go into the body and literally work there for a long time. And so this is more complex. And so that should have special considerations, right? So these are all questions that the FDA will be looking at and asking when they are evaluating your CMC section. All right, so now let's focus on the CMC section itself. Okay. And if you remember from last week, this is the figure 7.1 from the textbook. 7.1 from the textbook. And of course, today we are going to be focusing on module 3 right here, which is the quality section. That is where, if you remember, the CMC section resides in that module. Okay. Now, last week I told you that you don't have to memorize all these contents of the CMC section, right? But what I do want you to focus on and actually remember some of the stuff is you don't have to remember all the sections, but you should be able to recognize that these things are part of the CMC sections. That generally you're talking about manufacturing, characterization, control of the drug substance. So when you see a list like this, you should know that this is part of the CMC section. And so you can generally recognize manufacturer, stability. Right here is stability, container closure system, reference to standard material, reference to standards or materials, reference standards rather, or materials. These are all common parts of the CMC section. So you should be able to recognize these things as being part of the CMC section. Okay, and the other thing I want you to focus on is the difference between drug substance and the drug product. So what's the drug substance versus drug product? The drug substance is the active pharmaceutical ingredient. The drug substance is the active pharmaceutical ingredient. The stuff that you initially make from your fermentation and purification. At the end of purification, you essentially have the drug substance in a big, large container. So this is your drug substance. And the drug product is what the patient sees, right? It's the formulated version. So the drug product will look like a vial or a syringe, a small vial. Or a syringe, right? So that's the difference between drug substance and drug product. And you have to provide information on both of them in terms of how you manufacture it, how you characterize it, how you control the substance, etc. Okay, so I need you to be able to distinguish between the two, the DS and DP, right? Drug substance and drug product. Okay, so two things from this. Be able to recognize the general items in the CMC section and be able to distinguish between drug substance and drug product. Okay, so let's see what what needs to be placed in the CMC section. So what I have put here is a list of things that are included in the CTD format, the Common Technical Document. These are all the things that are listed in the CTD format, and I am kind of mapping it against what's required in the CGMP inspection. We will be going over this in a few weeks. It's listed in the 21 CFR Part 210 and 211. And what it is I'm just trying to show you how what's required of GMP is what's listed in the CTD. It's not a one-to-one correspondence, but if you look at, for example, manufacturing consistency, you have facilities and equipment listed over here, production control. So it's kind of a nice companion. They dovetail with each other, and they are like two companions that go hand in hand, right? So the CTD format, the CMC section of the CTD format, tries to address the GMP requirements, CGMP requirements, okay, that are listed in the regulations. Okay, so basically, I'm trying to show you how they dovetail with each other. Okay. All right, so these are all the regulatory requirements where CMC is needed, right? So you need to have a CMC section when you are filing an IND, and you need material from your pre-IND work to fill in the blanks for your CMC section for IND filing. And then you are going to need it again for NDA. And you want, how is post-approval involved in CMC? Post-approval. Why do we need to worry about CMC after your product has been approved? Anybody remember from last week? Something like 30-day change, 30-day change, 0-day change, what was it, CBE 0 and CBE 30. And then prior approval supplements, because you will probably change your process over time, and anytime you do change, you make a change to the CMC, you need to have communication with the FDA, right? And then I don't know if we discussed ANDA. ANDA is abbreviated new drug application. This is applicable mostly to generics. So when when a company is bringing a generic product online, they can submit an ANDA, and there again, CMC is required. Although for this generic section is mostly for small molecules, and there the process is not as important as the characterization of the product itself. Okay, so now, of course, with this does not apply. ANDA does not apply to large molecules. For large molecules, we will be you'll be dealing with what we call biosimilars, or what is also called follow-on biologics, right? So there the requirements are a lot more stringent than with small molecules. Okay, so the CMC section, what does it need to have? It should fully describe the composition, manufacture, and specifications of the drug product and of the drug substance. Remember the difference between product and substance, drug product and drug substance, including its physical and chemical characteristic and stability. Now, the requirements have been specified in 21 CFR 314. Okay, so you can Google that if you want, and you can, you will be able to find the details of the requirements for CMC. So basically, just Google 21 CFR 314. You should be able to find that. In addition to the requirements, the FDA often gives you guidance. So guidance for industry or points to consider. So these are documents that they put out, and even though they are not quote unquote set in stone, but those are expectations basically. Unless you can prove otherwise, the FDA expects you to follow the guidance. Okay. All right. So what else do we have in terms of guidance? Key guidance documents. Now, I don't expect you to memorize all these things, unless you want to be an FDA inspector. Of course, if you are an FDA inspector, you should be completely familiar with all of these. So the reason I have listed these is this is a good reference slide for you to know if you are dealing with biotechnology derived products. Then you should look up this guidance for industry, which was published in 1995. Then, of course, there is the guidance for industry for submission of CMC information for therapeutic recombinant derived product or monoclonal antibody for in vivo use, which was published in 1996. Then there is IND for phase 2 and 3 studies of drugs, published in 1999, well, draft. Again, therapeutic biotechnology derived products. So basically, these are all guidance documents that are relevant to biotech products. And these are, when you look through these, if you look through these, you will notice a lot of focus on the complexity of the products that I talked about earlier, right? So the focus of all these guidance documents is the much higher complexity of biological products. Okay. So there are points to consider in characterization of cell lines. For example, cell lines are living things, right? And they are derived from animal sources. So we really need to characterize them thoroughly. We'll talk about that in a minute. So these are the key guidance documents here. And these topics, by the way, are near and dear to the FDA. So when they look at your CMC submissions, they will be looking at it from these points of view, whatever is listed here. Okay. All right. So what does the CMC for biologicals contain? Well, it contains a description of the expression vector. For bacteria, it would be the plasmid. For animal cells, it would be the other, the viral vector that you are using. And of course, characterization of the cell banking system, the master cell bank, the MCB, or the manufacturer's working cell bank, um, and end of production cells. End of production cells are sample of cells taken from the fermenter after the production has been completed. And this is mostly to show that your cell line is stable. Because sometimes your recombinant cell lines may not be stable and it may actually lose plasmids over time. And so then the cells are not the same at the end as you started with, right? So you have to show cell line stability. Okay. Drug substance and manufacturing process and release, details of that. Same with the drug product formulation and release. Okay. So lots and lots of stuff that you have to show to the FDA. Okay. Source materials. You know that oftentimes the source materials are of biological origin. So for example, the genetic material, the genes that we are using, where did it come from? Okay. And how have you modified it? You know, you would cut the gene from the source material and then put it in a vector. So in the vector itself, expression vector, you need to provide the details of the origin of the construct, the plasmid map itself, for example, what are the restriction sites there, what are the integration sites, what promoter you are using, what's the copy number, etc., and the stability of the plasmid in the cell. So you have to provide details of all that. What sequence? You have to give the sequence of the gene itself. So you have to give the results of your gene sequencing data. Then the cells that you're using, if you're using bacterial or animal cells especially, where did you get the cells from? What is the history? So is there any potential of exposure to viruses? How you manipulated the cells to introduce the vector in it, and how you established the cell bank, whether you used, how you used cGMP protocols to establish the cell bank, how did you do cryopreservation, and of course testing of the raw material, source materials. The microbiology, is there any, did you do testing for that? Whether there are any viruses present, or did you test for viruses, and all the other tests that are listed here. Okay, so lots of characterization of the source material is needed because the source material is of biological nature. Other components, all components, raw materials, excipients, reagents, even though these reagents may not be present in the final product, as long as your product has seen them sometimes. For example, buffers are a good example of components that may not be present in so you might use one particular buffer during purification, and later on you will change it out to the final formulation buffer. But as long as that buffer has seen the product sometime during the process, you must describe it. Okay. So safety and quality of the material, source screening, the screening that you have done, testing. So again, all the materials that you provide, you have to evaluate in context of use, of course. Okay. So where did you use it, and what potential does it have to impact the product? Are there any known potential toxicities? So for example, we often use antibiotics in bacterial fermentation, and we use a material called methotrexate in CHO cells and other chemicals. So does it have toxicity, and have you addressed that? Usually you prefer to use products that have already been approved by the FDA. So for example, albumin is often used as carrier in the drug product, as a carrier in the drug product. So you use albumin along with your product. So since the FDA has already approved that in many products, it's easier to get approval if you use things that have already been approved. And you prefer to use clinical grade reagents whenever you can. Okay, but it may not always be possible. Okay, how are you guys doing? Still awake? Oh my God, everybody is asleep. Okay, okay. Oz and Tim are awake, and so is Jerry. Good, good, good. All right. Moving forward. Okay, a special concern is mad cow disease, right? Because why is this so important? Most of the mammalian cell processes use either serum, more specifically fetal calf serum, right? Fetal bovine serum, FBS. Either they use that, or nowadays people are using serum-free media, but even with serum-free media, they often use proteins that are derived from serum. Okay, so proteins that are derived from serum, and because of that, there is concern that you might get these prions that are really difficult to find, right? That come along with the bovine derived material, and it could cause problems later on, right? And so we have to make sure that the material that we get is from a country free of that disease. So people often source their bovine derived material from New Zealand, for example. I believe it is still free of mad cow disease. You want to identify the country of origin, tissue source. You want to basically make sure you know everything about where you got the material from, right? And maintain traceable records. But there is even though you might take all these precautions, you have to think, when is the mad cow disease going to go to New Zealand? It is possible, right? It is possible, even though we take all kinds of precautions. So really the best strategy is to avoid possibilities of TACs in the raw materials. So there is a lot of research going on protein-free media, for example. There is also, bovine serum, human serum albumin used in drug products. Nowadays, people are trying to go away from that because there is a possibility of viruses coming along there. Okay, manufacturing process and process controls. Again, I'll remind everybody that process is the product, so you have to make sure that you provide complete description covering source, expression methods, production, materials and components, etc., etc., etc. for manufacturing. Differences in manufacturing of drug substance and drug product in preclinical studies versus clinical studies. So you know that people in process development are always improving the process. And so the process that you used for preclinical studies is likely to be different from that you used for clinical studies. So you have to describe the differences, because oftentimes those differences might create differences in product. Okay. For example, you might have moved from a serum containing media to a serum free media, and that does affect the product sometimes. And you want to give a description of process controls. So what are the in-process controls that you have in effect? What is the temperature range? What is the pH range? Things like that. That could affect the safety. So for example, you have a virus inactivation step, cell irradiation steps to deactivate things. Okay, so you want to show data demonstrating safety-related processes are effective within the manufacturing controls. Okay, very important, viral clearance. This is particularly applicable to mammalian cell processes. Because there is always the potential of getting viruses into your product. So basically, what you have to do is that you have to show that your virus, any virus that may be in there. So you have to assume that there is virus in your product, and that your purification process is actually doing clearance of the virus, potential viruses. So the way we do that is we do a small scale clearance study. And so you basically actually spike the drug substance with a model virus. Okay, so you take a particular virus, you actually put a lot of that in your product, go through your purification process, and at the end of the process, you have to show that there is no virus in your final product. So this is how you validate, and you often validate several logs higher than what you might actually see. So you have to show several log clearance, several logs of clearance of viruses. And the way we get rid of the virus is we often use at least two different, orthogonal means different mechanism of actions to kill the virus. So for example, you might use low pH or nanofiltration or detergent treatment or heat treatment to kill the viruses. So if you use two different methods, that is usually sufficient to kill the virus and show that you have been able to clear the virus. Okay, now characterization. So when you have a product and you are submitting a CMC section, you have to characterize the product with multiple tests. And characterization that you send into the FDA is a lot more than you would normally do for lot release testing. Okay, so the product characterization is a lot more thorough than lot release testing. Okay, so multiple tests. So you have to provide the description of tests, analytical procedures, and acceptance criteria, and what testing you have done throughout manufacturing for the source material, component, in-process manufacturing, for the drug substance and drug product tests, stability, etc. and you have to show them what your appropriate acceptance criteria are. Now, in the early clinical trials, you may not have those criteria set up. Okay, so you have to report the results or give broad release criteria in early development, and then over time you have to establish the acceptance criteria for all different things. So more stuff on. So here are examples of testing that you need to do. So characterization, structural, physiochemical, and immunological characterization. For release testing, you have to do identity, purity, potency, strength, and safety. So for example, safety, you have to show limits of bioburden, that there are no or limited number of bacteria present, no endotoxins or limited endotoxins or process contaminants. How much protein you have in terms of strength. Is the protein that you're producing your protein or some other protein, right? And of course, purity is there. You have to create an impurity profile, so showing absence or limited impurities. Okay, and you have to submit test results with the available lots that you have. So at the IND stage, you might have lots from the preclinical studies, and that's what you submit to the FDA. All right, so differentiation between three things. There is complete product characterization. Complete product characterization you submit once to the FDA. Submit once, right, in your CMC section. Lot release testing you have to do with every lot that you produce. So lot release testing is a subset of the product characterization that you submitted, is a subset of that. So the number of assays that you do for product characterization is a lot more than the lot release testing, right? A lot more. So this is a lot more thorough. So basically this is an inverted pyramid. Product characterization, a lot more assays. Lot release testing, a subset of that, and then stability testing is something that you do on an ongoing basis. Only a subset of the lot release assay. Okay, so the number of assays is less for stability testing. This is ongoing all the time. Lot release, every lot you have to do certain tests and complete characterization that you provide in the CMC chemistry section. Okay, so lot release testing also includes the CMC control section and a subset of the chemistry section. So examples of characterization tests. So for example, you might submit reverse phase, size exclusion, ion exchange, HPLC tests, peptide mapping, mass spectrometry. You have all kinds of tests, carbohydrate analysis, lots and lots of tests. Of course, you don't have to remember all of these. Basically, you have to remember that characterization has a lot more testing than lot release testing, right? Lot release has fewer tests than characterization. So you have to do purity, identity, then protein content. Protein content you might do radio immunoassay, ELISA, UV absorbance, etc. And there is some additional expected testing. For example, you want to show that your cell banks are sterile. You don't have any contamination in there. And mycoplasma is important. Mycoplasma contamination. You don't want mycoplasma contamination, so you have to do testing for that, and you have to show absence of endotoxin. Okay, so you want sterility of cell banks, products, and the placebo when you are doing clinical trials. Right? Okay, so we talked about characterization. Now this is what you might do for lot release testing. So you remember this is a subset of the characterization testing, right? And so here you are showing tests for different things. You are showing tests for purity, identity, impurities, impurity profile, and safety, and strength, and potency, and quality. So these are all the different things that you're showing in your lot release testing. Okay. So for example, pH is a quality attribute, whereas this HPLC testing is usually a purity attribute. And isoelectric focusing may be an identity attribute. Okay, so these are all different types of tests that you need to submit for each. So this might be, this will also go with your certificate of analysis with your product, CFA. Okay, so that's what is included in the lot release testing. Now this is not an all-inclusive. Different products are going to have different numbers of tests that they use. So it depends on the product and the product class. Okay. And then further subset of those tests are the stability studies. So you want to show that your product is stable over a long time, because that's what determines the shelf life. Shelf life of your product, and the expiration dating is really important for you, right? Expiration dating, because the longer the expiration dating that the FDA approves, the better off you are. You can keep the product longer, right? So you try to get as long an expiration date as possible. And that approval of that expiration date really depends on your stability studies. Okay? So you want real-time as well as accelerated data, right? Now, one of the things that I have not mentioned in this slide is the stress studies. Stress studies. So, for example, have you tested stability under UV radiation? Right, and under light, under not normal light, and higher temperatures, etc. So stress studies are important. Also for transport and storage conditions. So for example, have you tried to transport the material under high temperature conditions? Because sometimes that can happen when you are shipping the material. The temperature might go higher than what you intended it to. So you should be doing stability testing under stress conditions as well. Okay. Another thing that goes into the CMC is the container closure compatibility. So of course the container is the vial, and the intent of the closure, which is this the stopper that goes in there, is to keep the bacteria away from your product, right? But when you use a different closure, you have to be worried about extractables and leachables. Okay, under normal conditions, could some things from the stopper leach into the product? or if it goes into a higher temperature setting, for example, you're autoclaving the vials, then something could be extracted from the rubber that is used in the stopper into the product, right? So you need to be worried about that, and you need to provide data about these two things to the FDA. Okay, so what are some of the challenges that we face when we submit CMC data? Okay, first of all, you have to show that you have been able to identify any and all impurities in the active products, and that you have that under control. Okay, sometimes you are not able to remove all the impurities, and it is not necessary to remove all impurities as long as your clinical trials show that with your impurity profile, your drug is safe. Okay, so as long as your impurities are under control, you're okay. Reproducibility in manufacturing the final product, so batch to batch reproducibility, that you have the same lot release criteria, that you can show that your product meets the same lot release criteria from one batch to another. Appropriate control tests. So you should have the test specified that what you're going to use. You have to think about this quite a bit, especially when you are doing INDs. You want to keep the specifications of in-process and final material as broad as possible. This is because you don't want to box yourself in. As long as you are satisfied that the product is pure enough for your clinical trials, if you keep the specifications broad, you will have better room for maneuvering later on. Okay, so it's important to think about these specifications ahead of time. Are you able to define critical manufacturing variables? A good example of this is cell number. Because cell number can vary from lot to lot, you want to specify a range of cell number that you're comfortable with, right? So these are challenges that you have to face. You know, how broad the specifications should be, what should be the critical, how do you define critical variables, how stable is your product. These are all challenges that you face when you are putting together the CMC section, especially for an IND. Because when you are doing NDA or BLA, you have characterized the process sufficiently. But for IND, there is often lack of scientific evidence to support your application. So you have to make do with what information you have, but at the same time, convince the FDA that your product is safe enough to go into human clinical trials, right? And as I mentioned before, sometimes you use different active materials at different stages of development. So again, you have to show that your product is equivalent. These are all challenges that we face in putting together CMC. What does the FDA do with your application? Well, they have a bunch of team members from all different areas. And if you look at the textbook, it tells you all the different sections that are reviewed. There is the pharmacology, there is the chemistry section, there is the clinical trial section, etc. Okay. So what does the CMC review include? These are all the things that the FDA looks at. All the different sections that the FDA looks at. And of course, the FDA then has to prepare a deficiency letter. If there is anything missing, then they prepare a deficiency letter and send you the letter saying you need to act on all these different things. Okay, so this is an important slide because what I have listed here are things that FDA has used for putting phase 1 on hold. So that means these are areas of concern to the FDA, and this, you don't want to have this stuff in your CMC. Okay, so incomplete description of the manufacturing process is commonly used by the FDA to put things on hold. Information on animal-derived components not provided. So, so this is basically lessons learned from the past. And it's a good checklist to make sure that your CMC section doesn't have these inadequacies, like inadequate demonstration of viral clearance, okay, and does not perform test for endotoxins at appropriate manufacturing stages. These are all things that you should know that the FDA is concerned about, and you should be concerned about that as well, not just because the FDA is concerned about it, but because it's important for safety of the patients, right? Inadequate sourcing and history of source material. Okay? All of these things are important. And remember, anytime we change the CMC, you have to notify the FDA. And remember the 3 different classes of change notification. All right. So this week's exercise, you are going to go to the FDA and evaluate the FDA's reviewer comments on one particular recently approved biotech product. So I'm going to quickly demonstrate to you how to do that. So I think I'm going to try to change to the web tour, so you should be able to see the web now, okay? And let me know if you don't see it. Oh, sorry, okay. All right, so do you see the web page now? It says enter a URL. So I'm going to enter www.fda.gov. Can you tell me if you see that? Are you able to see fda.gov? Okay, good. Okay, so from here you go to drugs, the drugs tab right there. Okay, so now I'm on the drugs tab. And in the drugs tab, I want you to scroll down, and you're going to go to the drug approvals and databases. So in the drug approvals and databases, I want you to go to the drugs at FDA search, and then your product will be listed under alphabetically. So I want to demonstrate Benlysta, so I will go to B, and under B I can go down, scroll down to Benlysta right here. Whoops, yeah, okay. I think yeah, that's the one. So I have the Benlysta under the product name. I can go to approval history, letters, reviewers, etc. And there I can go to the review section. Do you see the pop-up window that popped up? Okay, so I can't help you there. I have a pop-up window that shows all kinds of different PDF files, like approval letters, summary review, medical review, etc. And so I can click on the chemistry review. There is a chemistry reviews section, and there, when I click on that, it shows me the chemistry review for Benlysta application number 125370. And you can then review the whole document, and it goes into details about how Benlysta is manufactured, its characterization, etc. Okay, so so is that sufficient of a demonstration on how to get to those documents for your class exercise? So for your class exercise, you're going to select a different product, not Benlysta, but a different product, and then answer the questions in the exercise. Is that clear? I need a show of hands. Was everybody able to follow what I did? How I went to the Benlysta? Okay. All right. Good. Thanks. All right. So I think that's the end of the lecture. What questions do you have? Okay, so as a reminder, everybody should be doing 3 things every week. Number 1, attend this lecture and do the quiz. Now, if you do attend the lecture, you automatically get 80% on your participation. So the quiz is in lieu of the participation. Okay, and if you get 100% on the quiz, of course you get 100%. Right? So that's the first thing. Number 2, your assignment that you can find in the assignment. You can submit through the assignment Dropbox. You can find the assignment in the content section for this week. Okay. And number 3, discussion forum. So as long as you're doing all these 3 things, you should be doing very well in the class. Okay. And discussion forum, you need to do 3 posts, out of which 1 should be original. Okay. And the other 2 can be responses to other people's posts. So the more we discuss, the more we learn, right? And quiz and assignments, they're easy, usually easy. If you attend the lecture and you have your lecture notes with you, when you do the quiz, you should be doing well in it. and most people are doing well. Okay. All right, and you guys, since you attended the live lecture, you get the bonus of minimum 80%. Okay. All right, then, if there are no more questions, we'll stop there. Oh, it seems like Oz is writing something. Oh, okay. All right. Okay, then have a good weekend, and I'll see you either online or next week during the lecture. CC moderator, we are done. Thank you very much.